In today's lecture, we're going to make a transition. As we shift our focus to the second conceptual theme around which our course on the science of life is organized, the theme of development and homeostasis. Now, before we begin with this theme, I want to take some time to step back today and remind you of the overall organization of the course, and to look at the ground we've covered so far as a prelude for looking forward to where we're going to be going next. At the very beginning of the course, you'll recall, I introduced you to the hierarchical organization of life, with that hierarchy extending from very small things such as molecules and cells on the one hand, to the very largest and most inclusive units in biology, represented by ecosystems, or even by the biosphere of the entire planet. Now, the outline of a traditional introductory course in biology often is organized around a linear progression that moves through this biological hierarchy. That is, the course might start with some chemistry and then some molecular biology, move on to the structure and function of cells, then move on to organisms and looking at how they work, then move on to aspects of ecology and so on up the scale. This is a reasonable way to organize and present the many facts and details that you would encounter in an introductory biology course. But our approach in the science of life is a little different from this. Our purpose here is to try to bring out broad unifying principles of biology, principles that are useful for gaining an appreciation for the subject as a whole. For this reason, you'll recall, I've organized this course by dividing it into three major sections, each section centered on a unifying conceptual theme, a conceptual theme that I think provides a good framework for understanding the big picture that we want to achieve in biology. The first third of the course, the part we've just completed, was centered around the theme of information and evolution. And I'll recap the highlights of that section for you in just a moment. The second segment of the course, which we're about to begin, is going to be centered on the theme of development and homeostasis. As I'll explain in more detail later, in this part of the course, we're going to look at how complex living things get put together in the first place, how the various parts of a complex organism are coordinated with each other, and how the proper functioning of this complex organization is maintained in the face of internal and external challenges. In the third and final section of the course, we'll turn our attention to the theme of energy and resources, asking where all of the stuff we need to build and maintain living systems come from, and how we pay the energetic costs to do so. Within each of these three conceptual themes, you'll also recall, our approach is to explore that theme across the levels of organization in the biological hierarchy, considering each first from the fundamental level of molecules and cells, and then building our understanding of that theme up through progressively higher levels of organization. If we were to map the conceptual progression of the course then on a set of axes with time on the x-axis, that is the horizontal axis, and the level of biological organization that we're looking at on the y-axis, the vertical axis, it would look like a series of three sawteeth. Each sawtooth would represent one of the three main conceptual themes of the course. And within each theme, we begin at the molecular level and then move ourselves through progressively higher levels up to the levels of cells, organisms, populations of organisms, and so forth. As we begin each thematic section of the course, we drop down again to the lowest level of organization in this hierarchy and begin the progression all over, considering it now, however, from a different conceptual point of view. And that's where we are now, as we begin the middle third of the course, returning to the level of molecules and cells, but looking at them from the context of the theme of development and homeostasis. Now, before we move on, I want to take a few moments to remind you where we've been so far. We've covered a lot of ground, and so I think it's useful to look back over that terrain and review some of the key points we've addressed in the discussion of information and evolution. After we've taken this quick look backward, we're then going to look forward again, and I'll introduce the theme of development and homeostasis more formally, outlining where this theme will take us over the next series of lectures. So where have we been? We began our discussion of information and evolution in the first third of the course with the simple question of how life could have arisen from non-living matter on the primitive Earth. Now, this question is both simple, but of course it's very profound. And it brought us to consider the following key point. And that is this: in order for life to exist as we know it today, living things must be able to store and transmit information about their structure. Why? They need to do this so that they can make more of themselves. We learned that the critical information about structure that has to be stored and transmitted in this way is information about the structure of proteins. Now, we know that living things are actually composed of several broad classes of biological molecules, including carbohydrates, those are the sugars, lipids, those are the fats, nucleic acids, as well as proteins. But proteins are the molecular workhorses of the cell, and almost everything a cell, or actually really a living organism, does to be alive depends ultimately on a protein, or perhaps a collection of proteins working together, in order to get that job done. And remember also that to a first approximation, the way that a protein works has largely to do with the way that it's shaped. The structure and function of proteins has to do with their shape. And finally, remember that the shape of a protein, in turn, depends largely on its unique linear sequence of amino acids. So we know from that, therefore, that what we needed to do was find a way to code for sequences of amino acids. Now, of course, we know that the biomolecule that's responsible for storing and transmitting this information is the nucleic acid, DNA. We spent quite some time looking at the structure and function of this molecule, especially looking at the molecular mechanisms by which DNA is replicated, how DNA is transcribed into messenger RNA, and how that RNA intermediate, right, the messenger, is ultimately translated into a functional protein. A thorough understanding of DNA, of course, is essential for understanding how life works at all levels of organization. And I think it's fair to say that we've gained a fair amount of that understanding now. Another really important take-home message from this last section of the course is that as important as DNA is for living systems to work the way they do, it's actually helpless on its own. DNA may hold the code for building proteins, but it has absolutely no way it can do anything useful with that information on its own. Only with the help of a large and highly choreographed collection of proteins is that information, which is latent in a molecule of DNA, able to be turned into a useful form in terms of the structure of a protein. This relationship represents an essential catch-22 in biology. Proteins and other biomolecules that make up the structure of living things, such as ourselves, can't be built without the information stored in DNA. But DNA can't use this information without the help of proteins. How this molecular collaboration arose originally remains one of the key unsolved mysteries about the origin of life. Well, the next question we turned to was how DNA could be passed in an orderly fashion from a parent to its offspring. Here we began by looking at how the movements of chromosomes during mitosis, that process by which cells divide, those chromosomal movements ensure that both of the daughter cells following division have a complete complement of the genetic information that was possessed originally by their parent. We also looked at a special kind of cell division, you'll recall, one that occurs when cells divide in preparation for sexual reproduction, a process of division that we called meiosis. The way that chromosomes move and are divided up in meiosis led us to consider how these patterns of chromosome movement could account for the way that heritable traits, that is, traits that have some genetic basis, could be passed on from parents to their offspring in sexually reproducing organisms. And further, we saw how these patterns of trait transmission could be modeled by the conventions developed 150 years ago now by the monk Gregor Mendel. Mendel, you'll recall, inferred these patterns from his studies of plant breeding long before anyone even knew chromosomes existed. But it's the movements of chromosomes during meiosis that give rise to the way we see traits passed on from parents to offspring. Now, this work, combined with the understanding that we had previously of molecular genetics, led us to another very important observation. Mutations that arise in the form of errors when DNA is replicated, or as a consequence of damage done to DNA later on, along with the genetic recombination that we saw occurring as a consequence of the independent assortment of chromosomes during meiosis and crossing over during meiosis, means that genetic variation is constantly being introduced into populations of organisms. Offspring are rarely exactly the same as their parents. Although the DNA is passing on information to subsequent generations, that information is constantly being transformed in one way or another. And this variation, in turn, is the substrate on which evolution by natural selection works. Thus, the take-home message here is that evolution is the inevitable result of the way genetic information is replicated and transmitted across generations. And, thus, evolution is an inevitable property of life. Now, we looked in some detail at the mechanisms by which evolution occurs, beginning with the theory of natural selection first proposed by Charles Darwin. Arguably, Darwin's theory is one of the most important scientific insights of all time, on a par with other theories that have fundamentally altered the way we think about the world around us, such as Einstein's theory of relativity. Certainly, Darwin's theory completely revolutionized our understanding of the science of biology because it showed that the way things work, the way living organisms exist, has to do with history and organic evolution, not necessarily the design of the creator. This insight is important, of course, from a philosophical perspective, but we're not here to discuss philosophy. It's also important from a practical point of view, as we try to understand how living systems work. To quote Theodosius Dobzhansky, who was one of the principal contributors to the modern synthesis of evolutionary biology that we discussed, which occurred in the early part of the 20th century. To quote Dobzhansky, "Nothing in biology makes sense except in the light of evolution." Dobzhansky's point here was that if we try to understand how some biological entity works, we need to appreciate the fact that that entity is the result of an evolutionary process. Now, another point to reemphasize here, however, is that not all evolution is the result of natural selection as Darwin originally envisioned it. We saw also that genetic drift, which is essentially a random process, can be as strong an evolutionary force as natural selection, maybe more so in some instances. Thus, not all aspects of the way a particular biological system works is necessarily an adaptation. And by extension, we need to remember that evolution doesn't act to perfect organisms. Evolution is simply change over time. Natural selection leads to adaptation, but other evolutionary forces, such as genetic drift, just lead to change. Well, finally, we concluded our exploration of the theme of information and evolution by considering how speciation has led to the almost unfathomable diversification of organisms over evolutionary time that occurred over evolutionary time that we see in the world around us today. And we then asked how we could organize this incredible diversity and make some sense of it. Now, an important point to emphasize here is that ultimately, it's the information residing in DNA that underlies all of this diversity. It has to be. The structure and function of organisms ultimately has to reflect, in part, as we'll see as we move on later in the course, the DNA that that organism has. Now, there's an interesting consequence of this, and that is that it implies that the genetic information found in all of the innumerable organisms that exist today or ever existed represents nothing more than extensive modifications of the genetic information that was found in the very first single-celled organism that we would call living. This is a profound conclusion. And as far as we can tell at present, it seems as though it must inevitably be true. So, where do we go from here? How do we begin this next section of the course? Well, first, let me define the terms I've used to name the conceptual theme of this section. Development is a familiar word to us all, and obviously it has something to do with growth and change over time. Homeostasis is more technical, and you may not be familiar with this term, so let me give you a formal definition. Homeostasis is derived from two Greek roots, homos, meaning the same, and stasis, meaning standing or position. So homeostasis might mean something as like same position. The Random House Dictionary, the unabridged second edition, defines homeostasis as, and I'm quoting here, the tendency of a system, especially the physiological system of higher animals, to maintain internal stability owing to the coordinated response of its parts to any situation or stimulus tending to disturb its normal condition or function. Now, I realize this is a mouthful, but the points raised by this definition get to the heart of the conceptual issues that we're about to explore. To maintain internal stability through the coordinated response of different parts, along with the development of the system from which this maintenance and coordination must occur, are essential requirements for living systems at all levels of organization. To put it more simply, when biological systems began to have different parts, more than just one part, which actually had to happen immediately as soon as there were more than one kind of molecule getting together, as soon as there were different parts, there must have been some way for those different parts to interact in a coordinated fashion such that the interaction created a condition that benefited the whole more than the separate parts. In the broadest brushstrokes, this is going to be the central conceptual theme we're about to begin to explore. Now, I think a good way to begin to discuss this theme of development and homeostasis is by picking up where we left off in the last lecture, when we ended with a brief history of life on Earth, and we ended at the point where we had had the evolution of multicellular organisms, which was thought to occur about 1.5 billion years ago. Multicellular organisms are thought to have arisen several times independently. In other words, not all organisms today that are multicellular came from the same progenitor. In fact, the different broad kinds of organisms like plants and animals and fungi, so forth, that we see around us today, probably all evolved from independent events where multicellularity arose. We don't have any useful fossils or other kinds of definitive evidence to tell us exactly how multicellularity arose, but we can infer some things by looking at existing organisms. In all cases, multicellular organisms probably arose from colonial forms of single-celled organisms. Consider a group of green algae, present-day green algae, which are called the volvocales. Now, there are a whole number of different genera in this taxonomic lineage, some of which, such as members of the genus Chlamydomonas, are only found as single isolated cells. Others, such as members of the genus Gonium, occur in small bunches of cells. These cells don't occur in isolation. They always occur in small bunches of maybe 4 or 5, a dozen cells all stuck together. Still other parts of this lineage, such as the genus Pandorina, are found in even larger groups as hollow balls of cells. Now, in the most extreme case, in the genus Volvox, which is one of the favorite organisms to be used in high school biology because they're so much fun to look at, this organism is a hollow ball of cells like Pandorina, although much larger. There may be hundreds of cells forming this ball, and most importantly, some of the cells in this ball look distinctly different from other cells. Now, this group of algae, the volvocales, represents a particularly nice sequence of increasing colonial organization, from single-celled organisms to organisms that occur in very highly organized larger groups. And similar progressions can be found in other groups of organisms as well. But we shouldn't see this progression as actually being an evolutionary progression. These are all existing organisms today. I point this out as an example only because these existing organisms give us an idea of how an evolutionary progression might have occurred. Now here's the question. At what point in this progression should an organism be considered to be truly a multicellular organism? Gonium has multiple cells. Is it a multicellular organism, or is it just a colony? Well, there's no simple answer to this question, of course, but most biologists would agree that the dividing line is where different cells in the group start to have distinct functions and can no longer survive on their own. In Volvox, for example, a small number of cells in the ball are specialized for reproduction and only for reproduction. These reproductive cells obtain their nutrients and get other forms of support from the other cells in the ball. They can't exist on their own. Conversely, the other cells in the ball, the so-called vegetative cells, are incapable of reproducing. They need the reproductive cells to reproduce the whole group. Clearly then, Volvox is a multicellular organism, although about as simple a multicellular organism as we can imagine. But it's multicellular because there is a distinct specialization of cell types. And Volvox in this sense may not be too different, at least conceptually, from the first multicellular organisms to have evolved 1.5 billion years ago. Now, there were two key adaptations that must have occurred in order for multicellular organisms to have arisen at all. The first of these adaptations was simply the ability of cells to stick to each other. Most single-celled organisms, after they undergo cell division to reproduce, simply separate, and the two daughter cells go their separate ways. But an adaptation for single-celled organisms to sometimes not go their separate ways, to sometimes stick together, might have occurred if there was selection acting on some aspect of sticking together that made it better for the cells in that group to be together instead of apart. For example, a group of cells might have a larger mass, and if they're living in an aqueous environment, thus might be able to resist being pushed around by water currents if it's better for them to stay in one place. It might be harder for a clump of cells to be engulfed by some other cells that want to eat them as opposed to a single cell alone, and so forth. This kind of adaptation then would tend to make cells stick together, but what did they need to do to stick together? What they had to do was to evolve a more complex cell membrane, a cell membrane that included molecules, and these were probably proteins, that allowed those cells to stick together in a selective fashion, to stick together to another kind of cell of their own type. As we'll see later, cell surface molecules that allow cells to recognize each other and to stick together on the basis of this recognition are important, in fact essential, to many aspects of development and normal cell function in complex modern multicellular organisms. Now, the second of these key adaptations had to do with the ability of a single cell to change its structure and function. Why would it change its structure and function? Well, to avoid being killed if external conditions changed for the worse. For example, many present-day bacteria and other kinds of microorganisms can transform themselves from growing, reproducing cells into a dormant or resting form that we call a spore. In many cases, spore cells, the spore forms of these organisms, can resist unbelievably harsh conditions for extended periods of time, years, decades, maybe even centuries. The selective advantage of this trait, of course, is that the ability to produce spores allows these organisms to live in a broader range of environments than they could otherwise do, because those environments, if they happen to have harsh conditions, could be survived over some period of time by the spore of that organism. But what do you need to do to be able to change into a spore? Well, as we'll see, what a cell needs to do to change its function and structure is got to have something to do with the way that genetic mechanisms will change the proteins that are found in that cell. In other words, the ability of cells to produce spores must have involved genetic mechanisms for controlling DNA, which was a prerequisite for cells to become even more highly and permanently specialized as they are in modern complex multicellular organisms. Well, obviously, once cells are clumped in a group and can specialize, then you can imagine that selection could act on different parts of that clump in different ways. Cells on the outside might be specialized to resist desiccation, whereas cells on the inside might be specialized to process nutrients in different ways. And this increasing specialization, no doubt, had to be where the highly specialized different cell types that we find in complex organisms come from today. Now, as I mentioned in the last lecture, one of the first specializations, and one that we've just seen in the Volvox example, that must have occurred was a specialization for cells that would only reproduce. The advent of sex, you'll remember, probably actually greatly accelerated the rate at which evolution could occur because, as we've seen, sexual reproduction generates more genetic variation. But from our present perspective, any kind of cellular specialization raises a number of broad questions that provide the framework for the section of the course we're now just beginning. The first question is this: how can the progeny of a cell that's become specialized for sexual reproductive function alone, give rise to other kinds of specialized cell types? It's one kind of cell, it's the only one that reproduces, but somehow it, later on in the development of a new organism, has to give rise to a whole range of different kinds of cell types. This is the fundamental mystery of development, and one we're going to spend some time on. With the evolution of multicellularity, organisms evolved to become ever larger also, to include increasingly specialized kinds of cell types, and to organize those cell types into structurally more complex ways, such as into different tissues and organs. This increased complexity gives rise to a second broad question we need to think about. How can the different parts of an organism communicate with each other to coordinate their activities? Well, finally, the evolution of multicellularity also gave rise to an interesting new opportunity for multicellular organisms to explore in an evolutionary context. And this opportunity really brings us to the third major question we want to begin to address. If you think about a single cell, or even a small clump of cells, every cell is in direct contact with the outside environment. But as multicellular organisms became larger and more complex, most of the cells of the organism found themselves on the inside, and only a few specialized cells or tissues found themselves on the outside. That led to an opportunity. If the cells on the inside could be maintained in a controlled internal environment, an internal environment that was distinctly different from the external environment, then it would be possible for those organisms to inhabit a whole range of environments that they couldn't inhabit before. I mean, the simplest case would be for organisms to come out on land. Cells have to live in an aqueous environment. How do we live and other organisms live on land? Because we maintain a watery environment inside us. And we only do that because all of those cells are bathed in their own fluids that we control, we as an organism control. The question then is, how is it possible for organisms to create and maintain, within a very narrow range of conditions, these favorable internal environments for the cells that are on the inside? Well, by considering the evolution of multicellular organisms, we've now introduced the conceptual theme we're going to be exploring over the next 24 lectures. The first part of this theme is about development. How do we build a complex multicellular organism? The second part of this theme is about homeostasis. Once we have a complex multicellular organism, how are the activities and functions of the specialized parts coordinated in order to maintain a stable internal environment? To explore this, we're going to begin in the next couple of lectures to look at the molecular level of organization and to consider first how different kinds of cells of the same organism can express different proteins at different times or in different places. And as we'll see, this is the essential property that allows us to both develop and maintain multicellularity.